TWiM listeners, do you have science you want to share? Microworld.org is the best place to post your microbiology-related news articles, pictures, videos, papers, and more. Sign up for a free account at microworld.org/join and start reaching thousands of microbiology enthusiasts just like yourself.
This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 80, recorded on June 5th, 2014. Hey everybody, I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hi there, how you doing?
I'm well. Did you miss your TWiMless week last time?
I sure did. I'm sorry I missed that.
No, it's okay. You were traveling around New England, I understand.
That's right. I was gone from Boston where the ASM annual meeting, general meeting was held.
We did a little summary of what we thought was cool.
Yeah, I listened to you guys. You did all right. Not bad. Okay? Yes, sir. Not bad.
Also joining us today from southeastern Michigan, Michele Swanson.
Hello. Ann Arbor. Let's not be confusing.
Well, the thing is, and TWiV, I'd say where people are from geographically, then at the end of the show, say Ann Arbor. So I asked Kathy Spindler, where are you from? She said, southeastern Michigan. That's where you are, right?
It is. They're both true.
Okay. How are you?
Great.
You're always great, right?
Yeah, I feel especially good today. I uploaded an NIH proposal that I'm excited about yesterday. My teaching is done. I feel like summer is here.
Undergrads are gone, right?
Yep.
Ann Arbor is deserted.
Well, not exactly. We have a lot of festivals during the summer, so it's a great place to be year-round.
Must be a big difference, though. You have, how many, 40,000 undergraduates?
Something like that. Yeah, now the graduate students, many of them, of course, are still here.
Mm-hmm.
Although my daughter's a law student, and she is in New York City now for the summer.
Kidding.
Wow. What is she doing? Working? Playing?
She's working with the ACLU there.
Cool.
No kidding.
Does she play golf also?
She does not play golf.
Does that make you sad?
No. She's got a very active life.
Okay. Well, this morning, I just flew back from California, Elio.
Did you really? Where were you?
I was in San Francisco just for yesterday. I went in yesterday morning.
Wow.
And took the red eye back. I did 3 interviews for our textbook in San Francisco.
Neat.
Nice.
And it was Mike Bishop, Nobel laureate, Harmit Malik, and Arnie Burke.
Great.
Nice.
I'm having a lot of fun doing this. We're going to do 26 different interviews for the textbook. So I was in San Francisco, and it was windy and cool. And when I flew in this morning, it was pouring, and now it is nice. Weather's changed, so I'm very happy. All right, we have 2 very cool papers for your listening pleasure today. And I'll take the first one, which was just published in Science. Actually, you can't easily find the date here. Earlier this year.
April 2014.
Hey, Michele, look at all the authors on this
I know.
paper.
You're going to have to make an exception.
It's got so many. It must have 50.
It's more. 80. I counted 80.
You counted 80? Wow.
Yeah.
The name of the paper is Total Synthesis of a Functional Designer Eukaryotic Chromosome. The first author is Narayana Analuru. And then the last author is Srinivasan Chandrasegaran. And then the next to the last author is someone I know, Jeff Bookey.
Oh, yeah.
Who used to be at Hopkins for many years and now has moved to New York University right here. And actually, he gave a seminar on this very topic a couple of weeks ago, which I missed because I was at the GM, the ASM GM. No, that wasn't my excuse. Some other excuse. I always have a good excuse. And a lot of authors on here because this is incredible. I would say this is a tour de force of what they call synthetic biology. which is synthesizing things. Now, what they do here is they synthesize a yeast chromosome. So I figured this is microbiology, right?
Sure.
Saccharomyces, because I debated a little whether you guys would not want me to do this paper.
My yeast, my PhD was in yeast genetics, so I'm happy to see it.
Yeah, I figured you'd be okay with it. They are microbes, after all, even though they are eukaryotes, but every now and then we can do that. Yeast genome is 12 million base pairs, 16 chromosome, about 6,000 genes. Of course, the whole thing's been sequenced. And 5,000 of these genes are on their own non-essential. You can disrupt them. And you can actually get a library of all these individual disruptions. I have it in my freezer. Did you know that, Michele?
I didn't.
If you ever want it, you can have it, because I don't use it anymore. I bought it years ago for a project. It was quite expensive. They're all arrayed in, I think, 96 or larger well plates. Mm. But an interesting question is, how many of those can you take out at once and still have the yeast grow? And so what they do in this paper is kind of a step towards doing that sort of thing. The idea would be to flank each gene with sites for a nuclease so that you could cut it out. The nuclease sites are called LOX-P sites, and the enzyme that you would cut it out with is called the Cre recombinase. And there's a story there. Did you tell me that story, Michele, about why they called it Cre-Lox?
I did not.
Something to do with bagels and cream cheese.
Really?
Yeah, now I've forgotten. Someone will write in and tell us, yeah. Anyway, you can put these short sequences around any gene or anything in a genome, and then you can express the nuclease and have it cut out. So one of the things they do in this paper is to put these LOX-P sites around every one of the non-essential genes on this chromosome. Okay. So this is chromosome 3 that they decided to synthesize. The original chromosome is 316,377 base pairs. And they did this in 3 steps. And this is really interesting to go into this technology because it's really astounding. The first step, they assembled what they call building blocks. These are 750 base pair pieces of DNA. And they made those starting from oligonucleotides, which are from 60 to 79 bases long. And then they use polymerase chain reaction to assemble these oligonucleotides into the 750 base pair building blocks. And the cool thing here is that this was largely done by undergraduate students as part of a course at Johns Hopkins called Build a Genome.
Wow.
Isn't that great?
That's very good.
I remember when I was in college, I went to Cornell, I took a genetics course taught by Jerry Fink.
Ooh.
Who you would know, being a yeast person, right? And he used to work on histidine, the histidine operon. So he had, in the lab, we all used to make mutants and isolate auxotrophic mutants, and he wanted all the histidine auxotrophs that we isolated. Because he would then look at them and see if they were interesting. So it's good to involve them. So this is really a massive thing. They had lots and lots of building blocks. So that's the first step. Then then there were 133 building blocks to the left of the centromere and 234 building blocks to the right. And they assembled those into what they call mini chunks of 2 to 4 kilobases. They did 44 and 83 overlapping mini chunks. So now they took the smaller building blocks and they built them into bigger mini chunks, 2 to 4 kb. And these mini chunks were designed in a way so that they overlapped by one building block, which is 750 base pairs. So they could put them into yeast by DNA transformation and get them to assemble by homologous recombination. Because that's something yeast do well, right, Michele?
They sure do.
They love to do homologous. So they would put a number of these mini chunks and they would be assembled in yeast into longer pieces. And then finally, they would put pools of mini chunks into yeast, like about 12 mini chunks at a time. They would introduce them into yeast and apply the appropriate selection and replace the corresponding regions of the chromosome 3 in the yeast. They had to do this.
The beauty of this is that it's all done in the proper order, right?
Right.
The species cannot assemble unless they're in the right order.
Right. And they did this 11 times. So 12 pools 11 times to get the entire yeast chromosome 3 replaced with their synthetic one. So, right, you have to do this all in the right order. And then they verify that. So they call this new chromosome SYN3, SYN3. And they can do gels, pulsed field electrophoresis, where you can look at very big DNAs and you can see this was slightly shorter than the regular chromosome 3. They also sequenced the whole thing. So this synthetic or designer chromosome, as they call it, is smaller. It's 272,907 base pairs compared to 316,000. And they designed the whole thing. Their goal was to make a chromosome that would confer wild-type growth and fitness to the yeast. But they took out a number of sequences that they called destabilizing elements, and then they put in the LOXP sites around the genes. So the destabilizing elements are things like tRNA genes, transposons, sequences around the telomeres, introns, some of the mating loci that are silent. They took those out, and then they put the...
Why are they all destabilizing? Is that clear to you?
Maybe Michele knows.
I don't know.
I think that these are areas where you see, deletions or recombination in the genome particularly, and so they just wanted to remove that so there wouldn't be any effect on their chromosome. But I don't really know for sure. So then they add PCR tags here and there so you could tell that you had put in a synthetic chromosome. And then, of course, the LOXP sites. And then they changed all the TAG stop codons to TAA. Every one of them. I don't know how many there were.
Wow.
And that way they say it frees up the TAG codon for future genetic expansion. So you could use that. So there's no more TAGs, so you could use that in the future for something else. In all, they made 5,410 base changes. And this synthesis is really a big deal because every building block and mini chunk and all that, they all had to be named something really precise. They had to track everything to make sure there were no errors.
The bookkeeping is amazing.
Yeah, the bookkeeping alone. And of course, you're getting all this stuff from undergraduates. You have to check it to make sure it's right.
Right.
So this is just, that's why there's so many people on this paper. I guess a lot of those are the undergraduates,
But
too.
how nice that the undergrads got a science paper.
It's great. It's just amazing. So this is one chromosome. I mean, eventually people want to make entire organisms,
It's
right?
It's the tiniest chromosome of the yeast, isn't it?
Yeah. So, boy, what an amount of work. But, this is all possible now because technology and DNA synthesis is so cheap. So that it's a tour de force. I think this is amazing.
And they were motivated because it's proof of principle, or they demonstrated some utility?
So it was mainly proof of principle, and also I think putting the LOXP around all the non-essential genes. They just wanted to make sure that didn't confer any phenotype, because as you know, if you put LOXP around every non-essential gene, and then they all come out at once when you put the recombinant, so that's not useful. So I think in the future they'll have individual chromosomes where you have one gene flanked in 2 and 3 and 4 in different combinations, but this way, at least they know that having LOXP everywhere is not a problem. So they sequenced this yeast strain containing this synthetic chromosome. They found a few mutations in the sequence, I think 9 or 10. Most of them are of no consequence. They think some of them are due to the fact that homologous recombination, which they use to assemble this chromosome, is error-prone. Is that true, Michele? I'm digging back in your history there.
You are. And I was actually reading a paragraph in the discussion, so I didn't hear the question, Professor.
That's okay. At least you admit it. You could have made up an excuse. Is homologous recombination error-prone?
I mean, it depends on what rate you would consider prone.
Yeah, any kind of mutation, I guess. They did, a number of overlaps, and they got, I think, 4 in the whole chromosome. So it's not very much. Anyway, that's what they say. It's error-prone. And then one of their LOXP sites wasn't there. They expected it to be there, so that's another mutation. They checked colony size, growth curve, and morphology under different growth conditions, different temperatures, different media, and the only one condition, which was high sorbitol, showed a slight fitness defect. Others, nothing. So this Again, this is a strain with a synthetic chromosome 3, and as far as I can tell, everything is fine in growth and culture.
Do you remember what kind of medium they used? I didn't look that up.
Oh, they used many different kinds. You mean what the basal medium?
For comparing the growth rate of the different strains, or the two strains.
Yeah, they used a variety of different media. I can pull that up here. It's all in the supplementary data, which goes on for many pages. Let's see. The synthesis takes many, many pages on its own.
But in figure 3, when they're looking at various temperatures, they're using a standard rich media, yeast extract peptone with sugar.
Right.
Yeah, and they have a variety of additions and subtractions, which, for example, different temperatures, 30, 37, YPD, yeast peptone something, is that what that is?
Yeah, dextrose. That's a rich
one.
complete media.
Then they add camptothecin, sorbitol, hydroxyurea. I guess MMS is the mutagen, benomyl, YPGE, then low pH and high pH cyclohexamide. And so those are additions to the media, but none of those had any effect. So they were all added to the rich media.
It's really very good. I mean, I enjoyed hearing this because often the idea of declaring that something is an essential gene doesn't take into account the environment. You know, it's essential when, essential where. So they did a lot of this.
It's also, they passed it for 125 generations. And the stick chromosome is stable. They have 30 different lineages that they checked in this way, 125 generation Gs, and they can use these PCR tags that they put in and show that there's no deletions or insertions or rearrangements. So this chromosome is functional and it's replicating clearly.
I guess the only thing they didn't do is to use it to make some beer.
That's right.
Or bread or wine.
Or bread or wine. See if it works just as well. But probably
then
does.
People would say that it's genetically modified and they wouldn't consume it. Right? You couldn't sell it. They made a haploid strain with the synthetic chromosome. And then, of course, they introduced Cre recombinase in an inducible manner, and that's lethal because, of course, you're chopping up most of that chromosome 3. In a diploid strain where you have 2 copies of the SYN3 chromosome, producing the Cre recombinase results in some gene loss, which you would expect. So these lock sites are functional. So basically, they conclude that the yeast genome is pretty pliable because you can modify it by thousands of bases as they've done here. And this is the basis for future modification. So you learn here, as Michele said, this is a proof of principle. I guess if you want to know if different genes can be taken out in combination, you start with 1 and 2 and 3. I mean, that's a lot of work, obviously, but that's what they want to do. I suppose they're going to make other synthetic chromosomes in the yeast as well and just maybe make one strain which has all of it synthetic. So that's that. I think that was pretty cool.
I wonder how long it took.
That's a good question. I have no sense for how long. You can't really tell. Probably a couple of years, I would guess.
Yeah, I don't recognize any of these names as now tenured professors. So it's not like the students did it 20 years ago.
Although you could argue that doing this work as an undergraduate is almost a sure path.
Yeah, I'll email Jeff, actually, and ask him and see how long it took. They dedicate this paper in the acknowledgments to the memory of Har Gobind Khurana, who synthesized the first yeast tRNA gene.
That's right.
Many years ago.
Yes.
Who passed away not too long ago, I think, right?
Not long ago. That's right.
Yep.
Yeah. It's wonderful that they did it because Gobind was just a phenomenon. He was a force of nature.
You must have known him. He was in Boston, right?
Well, I did a sabbatical with him.
Wow, you really did know him.
A full blast sabbatical, and I spent the year playing around. And we were working on synthetic fatty acids that could be used as markers for eventually for studying protein-lipid interactions. He didn't get that far with it, but it was a lot of fun. So I had a great time with him. But I tell you, nobody was more dedicated in science than Corona.
Yeah.
Amazing.
Can you imagine? It probably took him years to make a tRNA, right?
Yes, it did.
Look at this in comparison.
I know
what you can do.
He didn't use 85 people either.
No,
he did it himself.
I've always thought that in yeast, you're only limited by your imagination. The tools they have available in that system are just amazing.
Why did you not stay in yeast, Michele?
Well, I sort of got the infectious disease bug. I wanted to take the genetics that I'd learned in molecular biology and apply it to something that felt relevant to human health, more closely relevant.
I understand that, yeah. But yeast is cool, though. You can do a lot of things.
It's very cool. Yeah, I've always thought I'd get back to it, but...
Now, you were in Marion Carlson's lab, right?
I started there, and then I transferred to Fred Winston's lab, where I did most of my PhD. But both Marion and Fred were authors on my first paper.
Huh.
Well, the second paper we have today is not yeast. It's right up your human pathogen alley, right?
That's right. And it is titled... Retracing the Evolutionary Path That Led to Fleaborn Transmission of Yersinia pestis. It was published in May in the journal Cell Host and Microbe, and it's a paper from the Rocky Mountain Lab, which is a branch of the National Institutes of Health out in Hamilton, Montana, by the group of Joe Hinnebusch, and the authors are Yicheng Sun, Clayton Jarrett, Christopher Bozio, and Joe Hinnebusch himself.
Michele, do you know Joe?
I do. And actually, I met Yicheng Sun at a conference in Colorado 5 years ago.
So is he a middle or late stage investigator? I don't know him at all.
Joe?
Yeah.
He's a senior investigator.
Senior investigator, okay. I'm going there in a couple of weeks.
Oh, it's beautiful. This is the time of year to go.
I was invited to give a seminar, and I'm doing a TWiM while I'm there.
Hi.
They're going to show me the BSL-4 lab.
From the outside or in the inside?
They're going to I guess they'll bring me in, yeah.
Wow.
Our listeners might be wondering why there is an NIH lab out in the mountains of Colorado, but it is for historical reasons. There was an outbreak of Rocky Mountain spotted fever and needed to be studied on site. And ever since, there's been this small, highly productive group there studying lots of different infectious diseases.
And I'm told it's like one of the most beautiful fishing places in the world.
It is, and they're proud of it. There's a lot of hiking, skiing, you name it.
So when I'm there, David Baltimore and Alice Wang will be there. David will be fishing with Irv Weissman.
Nice.
And I understand that Stan Falco fishes there as well.
He has a home in that
there
region.
all the time. He flies there, I think.
Yeah.
Yeah, David has a house too. So all these microbiologists that like to fish. I don't fish, so I don't go there.
Yeah, it's trout fishing, I think, that's especially good.
Right. Okay. Sorry to interrupt. I just had to tell you that I was going there.
Quite all right. So most of our listeners have probably heard of Pestis because it was the cause of bubonic plague, this pandemics that swept across continents and killed millions of people at a time. So Black Death, it's known as bubonic plague. For example, in the early 1300s, there were 30 million deaths in Europe due to this infectious agent. But what's interesting is it's a close relative to another species called Yersinia pseudotuberculosis, which I'll probably refer to as pseudo-TB in this presentation. And pseudo-TB is, by comparison, a very well-behaved pathogen. It's acquired by eating contaminated food or drinking contaminated water, and usually it stays well contained in the GI tract, so you might get some diarrhea or some discomfort in your abdominal region, and then it's shed in feces. So the question they wanted to tackle is how is it? So once they sequenced both Yersinia pseudotuberculosis and Yersinia pestis, it was clear that even though the pathogenesis and the life cycle of these two bacteria is really quite different, their genomes are remarkably similar. So it gave them an opportunity to ask, using molecular genetic techniques, how did Yersinia pestis evolve from its progenitor pseudotuberculosis? So let me set the stage by describing a little bit about rodents and fleas. So pseudo-TB, as I said, it's an environmental bug that is acquired by animals in food, and it passes through the rodent, can replicate somewhat in the GI tract, and then is shed. So those same rodents, let's imagine a rat or a prairie dog, have their own companions in the flea world. So imagine Linus in Charlie Brown, who's walking around with the dust cloud. So rodents will have a flea community that lives with them, and the fleas suck blood from the rodent, from the rat, and then can feed on another rat in the area. So the question is, how did pseudo-TB, who is this normally well-behaved GI pathogen in rats, acquire the ability to be spread by fleas, which is what Pestis can do? And so the way they tackle this is to make some deductions from what they had learned about the biology of Pestis and pseudo-TB, and actually engineer strains where they essentially swapped a very small number of factors and could engineer in the laboratory a pseudo-TB that behaved like Pestis and a Pestis that resembled pseudo-TB. So that's where this story is going. So let's see. The first thing you should know is that bacteria can acquire genes by horizontal transmission. So they can acquire large plasmids that have new traits encoded on them. So one of the first changes that occurred between pseudo-TB and Pestis was the acquisition of a large plasmid that had a particular enzyme, phospholipase D, that allows the bacteria to survive in the midgut of fleas. So without this gene, if a flea feeds on a rodent and takes pseudo-TB into their bloodstream, the pseudo-TB will pass all the way to the hindgut of the flea and just be shed in the feces. But once they acquired this plasmid that encoded the enzyme that allowed them to survive in the midgut, now there were fewer competitors for that new strain, and they were able to multiply to higher levels. So now we have fleas that have more pseudo-TB in the midgut. And then through loss of function mutations, the bacteria acquired the ability to accumulate high levels of a small signaling molecule called cyclic di-GMP. And it's known from studies of a lot of bacteria that cyclic di-GMP is very good at recruiting a developmental pathway called biofilm formation, something we've talked about on this show.
You want to spell out what it is?
Cyclic di-GMP? Go ahead, Elio.
You do it.
It's a derivative of a guanine nucleotide that acts as a second messenger in bacteria. So when the levels of cyclic di-GMP are high, the bacteria can now form a biofilm. So the consequence of that for this evolving bacteria is that now, once they were growing to high numbers in the midgut, some of them then were able to latch on to a valve that is in fleas between their esophagus and their stomach. This valve is called the proventriculus, and these bacteria that were now making a lot of cyclic di-GMP could form biofilms, which are adherent, stable communities on this valve. So the consequence for the flea is that when it ingested a bacterium that can bind and form a biofilm on its valve in its esophagus, is that while that flea is feeding, the bacteria have grown to large enough numbers that they're actually blocking the pathway through the flea, and as a result, the flea becomes blocked, and they actually regurgitate blood into the animal that they're feeding on. And in that blood meal, they will also regurgitate some of the bacteria. So now
Wait a minute, hold on. The regurgitation is because they're trying to unplug that site, right?
It's just a physical response,
Right.
is my understanding. Right. So that this biofilm results in regurgitation of the blood during the time when the flea is feeding on a rodent, and therefore introduces some of the bacteria into that rodent. So
the way I see it is that the plug that's there bothers the insect, and the insect's trying to get rid of it.
Right. And some of the bacteria then get washed up with that in that blood meal and transferred. So now we've gone from a bacterium that is restricted just to the GI tract and the lower GI tract to a bug that can now be spit into the bloodstream of a rodent. And this allowed Yersinia pestis then to be spread by a flea vector and completely change its ecological niche. So the way the group tested this idea and established it was using molecular genetics. They essentially started with a wild-type Yersinia pseudo-TB. They started with a wild-type Yersinia pestis, and they stepwise swapped each of these 4 genes so that they engineered gradually in stepwise fashion pseudo-TB to look more and more and more like pestis, and for pestis to lose each of these functions and look more like pseudo-TB. And then the assays that they used were to allow the fleas to feed on blood that contained different variants of the bacteria, and then to dissect out their GI tract and look where the bacteria are. And they were able to do this because they had labeled the bacteria with green fluorescent protein. So you can beautifully see in their illustrations where the bacteria are located. So for example, the Yersinia pseudotuberculosis, after they've been eaten by a flea, are concentrated way at the tail end, the hindgut, whereas the pestis are concentrated at the closer to the mouth, the esophagus, this paraventriculus valve. And then the stepwise mutants that are intermediates, you can see they're thriving in the midgut.
They're really amazing pictures, aren't they? Really gorgeous.
Beautiful. And they also put numbers on that so they could then quantify how many fleas were infected, how many bugs were in each flea, and could show this gradual shift from pseudo-TB into a more pestis-like varmint. And then they did the ultimate experiment, which was to ask whether this change in location within the flea had an effect on the flea's ability to transmit the bacteria from one rodent to another. So again, they used their blood meal model, allowed the fleas to feed on different variants of the bacteria, and then put those fleas on another rodent and monitored its ability to transmit the bacteria to another rodent. And in a beautiful set of data in their 4th figure, they show that their engineered pseudotuberculosis is now 10,000 times more transmissible. You get 10,000 times more colony-forming units transmitted by those fleas than by the wild-type pseudotuberculosis. So it's mimicking Yersinia pestis after just 4 mutations, 3 of which affect this production of cyclic diGMP, and then the other gave it the ability to survive in the midgut.
Do we understand why that gut survival is engendered by, what was it, a phospholipase C?
Phospholipase D. I did not look back, and so I don't know if that's understood. But they describe that as, in evolutionary terms, as the key innovation. So it was the first key step that gave the Yersinia access to this new niche, allowed them to grow to higher numbers, and then, of course, that gave evolution a larger playground, more chance to acquire these other favorable mutations.
And that plasmid was acquired probably randomly.
Horizontally.
It's in the soil, and it just acquired it from some other source, right?
Right. So they then, to see if this model is feasible, they took advantage of a large amount of sequence data that's available for Yersinia strains isolated from all over the world at different time points, and could show, using a phylogenetic argument, that these events, same genetic events, have occurred out in nature. So it's really a beautiful example of evolution. And I think for any faculty that are listening and looking for a good model system to teach concepts in evolution, this is a really great example of how it can happen in a stepwise fashion.
It's amazing. It's just a great story.
Yeah.
Don't you think that it's a little bit counterintuitive that the great events were loss of function instead of gain of function?
Yeah, although they're easier. It's easier to get loss of function, so you might think that would be more frequent.
Sure.
But
still, it's sort of counterintuitive, isn't it? I mean, you want something bad to happen.
Since
you do it by losing function instead of gaining function.
Yeah, but what they lost was a repressor.
Yeah, I know.
So
I understand that.
They gained function, and then they lost two enzymes.
It takes a while to swallow that, in a way.
Is that a pun?
No, I didn't mean that. Oh, sorry.
So I'd like to just say a little bit about the first author, Yiching Sung. He actually did his PhD in Peking University, and then came to the U.S. for his first postdoc in Craig Darby's lab, where he started to work on Yersinia pestis and this particular regulator of biofilm formation that was relevant here, the RCSA gene. So he published a couple papers, actually in collaboration with Joe Hinnebusch, and then moved to Joe's lab to do this evolutionary project and test the ideas that they generated with Craig Darby. And Yiching Sun is now has his own lab at the Institute of Pathogen Biology in the Chinese Academy of Medical Sciences and Peking Union Medical College, where his lab is continuing to study biofilm formation.
Does he study Pestis still or something else?
I don't know.
That's pretty neat.
Know.
I guess it's a big change from Hamilton, Montana.
Sure is. Of course, Hamilton is a big change from San Francisco, too. So, as you'll see.
As I will see. I've never been there, yeah. So I think what is another interesting point here is that they say in the discussion that these are probably the major differences, but there are probably some minor ones as well. For example, why Pestis can't colonize the flea hindgut any longer.
Right.
Whereas why PT does. And they say it's probably because the bacterium lost an adhesin, right, which would allow it to do. They also make the interesting parenthetical comment, growing in the flea hindgut would be a dead-end sink for Y. pestis now. So I was trying to figure out why that would be. I guess, so in the gut, it would pass out and be in the soil, but it's not going to be transmitted that way anymore, right? That's why it would be a dead end?
It may have lost. I think I read a little bit about the genomics of these two bugs, and I think that about 17% of the genome of Y. pestis is now pseudogenes. So they are slowly losing functions that were important when they were pseudo-TB and living in the GI tract, but now they're not under selective pressure, so they're being lost.
So it's no longer a soil-dwelling bacterium; it's dwelling in the rodent and the flea hosts, right?
Yeah, I was trying to think about how to describe these evolutionary principles to a general audience, and the best story I could come up with is, imagine whose family business is to sell hot dogs on Coney Island from a cart. Where there's a lot of competition. But that person at random is taking courses, and they take a course in French, and they learn to speak French. So they then travel to France and are visiting the beaches and find no hot dog vendors. So now their hot dog vending skills have a whole new niche, and that family for generations, is now famous for their hot dogs on the shores of France, but they lose the ability. They lose the ability to speak English, so they can't go back to the Coney Island.
That's great.
So adaptive traits, and then you lose traits that you're not using. Use it or lose it.
It's nice.
It's the best I could
Assuming
do.
no one speaks French in Coney Island. Is that how you would say Elio chien chaud?
I guess.
I don't know if they actually say that. This is great because they could actually track this story. I don't know for any other pathogen that you could do this, right? Certainly.
Right. Plus, they can manipulate it easily, so they could do the experiments. Yeah, it's a beautiful system. But I thought, too, that, Vincent, you might want to make parallels to evolution of flu. There's a lot of worry about that every year.
So, we believe that pandemic strains of influenza arise from viruses that are circulating in aquatic birds, and we don't really have the nice succession as you do here, where you have the strain in birds and then you can see it in people and what changes it had to undergo in order to make that jump. So the biggest one is 1918, which killed millions of people globally, of course, and that virus is thought to originate from a bird, but we don't have the bird virus. As we do here, we have YTB and YPT and Y pestis. And so that's why this is beautiful. We don't have that pairing for any virus, really. I guess the closest thing would be SARS, perhaps, where we have some strains in bats and civets, which are different from the virus that infected people. So you assume that those changes were important, but of course you can't do the experiment to prove that. Here you have the natural host of this. Mm-hmm. So I
believe there are labs now doing this kind of research, though, testing in real time viral evolution and host switching.
Sure, with flu you can do that because so many strains are isolated every year, and you can sequence them all.
But animal models too are available, right?
We do have them, but they're not. They're animals.
Well, not for flu, but for other viruses.
Yeah. I don't know yet of any beautiful story like this one.
Yeah, not yet.
So Michele, then the idea is this. Y pestis arose that could then transmit via fleas, and then that got into people because people were living with rats, and they brought their fleas with them, and they bit people, right?
Yeah, and that's accidental. I mean, the pestis will do fine without people. It can go from rodent to rodent.
So pestis still exists out there in nature, right?
It does, and here in the U.S., they still get about estimated 1,500 cases a year, mostly in the Southwest, and it's prairie dogs that are infected, and then fleas that feed on the prairie dogs can spread it among prairie dogs, but also tourists and people that live there.
And we don't have a big rat problem? Is that why we don't have?
That's right. There used to be more pestis in cities, but then we did a better job of keeping the rat populations under control. So less disease in cities.
Although I must tell you that here, as you know, in New York City, we have them in the subways.
Yeah, but can the fleas survive in the subways?
I don't know. I have no idea. But people are looking at the, I think Ian Lipkin is looking at what's in rats in the subways. He's sequencing them. So we'll see I bet if he finds some Y pestis, that would be scary. He might, right?
Yeah, evidently. One reason this is such a bad bug is it only takes 100 to 500 bacteria to cause disease, and you can die within 24 hours if you're not treated. So it can get into the bloodstream, and things go south in a hurry.
These experiments at Rocky Mountain, are they done in the BSL-4 lab? I don't know if it's a 4 or a 3 pathogen.
Yeah, I don't know. I don't know.
I will ask Joe when I'm out there. All right, thank you, Michele.
My pleasure.
Did you like that paper?
I loved it.
Yeah, I figured you would.
It's very cool.
And we hit both of your major interests today, yeast and a bacterial pathogen, right?
And we talked a little about golf at the beginning, so
you have to be happy, right?
It's a good day.
But I'm ready to take a trip to France to savor the hot dogs on the beaches there.
And they've probably changed a little too, right, Michele, when he makes them in France?
That's right. They're probably not topped with mustard Well, maybe they're mustard. I don't know.
Something else, French
maybe
mustard.
they're on croissants.
Some people would probably get mad at that thought, but it sounds good. All right, let me read a few email here. We have lots. It's great. First one is from Bob, who writes, Dear TWiM hosts, I enjoyed episode 76, Genetic Biopixels in a Pathogenic Sweet Tooth. I really enjoyed hearing about the course that Dr. Schaechter teaches, and in particular the work his students did in developing the biosensor. I would like to recommend a couple of papers that a classmate in my differential equations class told me about. They can be found at Biomed Central and are open source, which is great. The first is Solving a Hamiltonian Path Problem with a Bacterial Computer by Baumgardner et al., Journal of Biological Engineering. The second was also published in the same journal, Engineering Bacteria to Solve the Burnt Pancake Problem by Haynes et al.
Oh, boy. Sounds great. Thank you very much.
That's you, Elio, right? You did this. You taught. You had students and developed a biosensor. Is that right?
Students who came from that lab. Yeah. Okay. Just how much they did themselves, I've forgotten, but they were part of the lab, and they're continuing. So it's an ongoing set of projects. Sure, great stuff. But really, what this illustrates is how penetrating it has gotten to be, that this biommathematics is really a field that just has come of its own.
So he explains a Hamiltonian path problem involves finding a root in a directed graph that starts at a node, the beginning node, and visits all of the nodes in the graph exactly one time. Companies like FedEx solve these kinds of problems daily in determining the most economical and efficient routes for their delivery persons. Solutions to these types of problems are very complex and computationally intensive. The authors used E. coli containing a HinXC recombination system from Salmonella typhimurium to randomly shuffle DNA segments as the computing system. They represented nodes in the graph as linked halves of two different genes encoding red or green fluorescent proteins. The bacterial populations displayed phenotypes that reflected random ordering of the edges of the graph. A Hamiltonian path was reported by fluorescing both red and green, resulting in yellow colonies. The burnt pancake problem involves sorting a stack of distinct objects into proper order and orientation using the minimum number of manipulations. This paper describes a proof of concept of in vivo computation. I enjoy so listening much to all of TWiM, and TWIP podcasts. Since I've been working on my math degree, I don't really have anyone to talk biology, biochemistry, etc. with, and I miss it. I'm down to the last two courses before I'm finished, real analysis and probability. I'm hoping that I can find a lab to volunteer in to help out. I've been looking so far, but no one has been keen on having a 70-year-old guy hanging out in the lab.
Oh, my goodness. Wow.
Oh, well, I have been able to take some interesting breath classes and just finished in bioinformatics. It was a trip to actually do sequence analysis and construct heat maps and run blasts. Anyone working on a machine to reverse aging, I'm a willing volunteer. Weather in Orange County this week has been hot and dry. Today was 98 Fahrenheit, 4% humidity, dew point 4 degrees, winds 20 miles per hour. We have more of the same predicted for tomorrow and into the weekend. Too windy to sail, too hot to just lay around at the beach, and with the winds, the surf sucks. So good time to hole up inside the air conditioning and enjoy some episodes of TWiM. So Bob is a retired biochemist, faculty member, I forgot where he was, who obviously likes outdoor stuff, but still is keeping himself educated.
That he should get into higher mathematics at this point in life is totally admirable.
It's
Bob,
great.
Thank you for showing us how it's done.
Why don't you let him work in your lab, Elio?
Well, it'd be good if I had a lab.
Next one is from Katie, who writes, Dear TWiM team, I'm currently studying for my biology degree, second-year exams in the UK, and I've listened to my first TWiM podcast today. I'd like to quickly thank you, as the zombie plant topic made perfect outside reading for my plant exam in a few days. I'm about to tackle the paper now. I will continue to listen for more interesting topics. Thanks so much. Pleasure. And Mark writes, I'm giving a final exam right now. This, by the way, Elio, is Mark Martin.
Oh, my.
And Michele, right. You both know him.
Yeah, we met him at ASMQ.
Then he said, I'm flying out to Boston for ASMQ ASM. Maybe I will see some of you there. I think he saw all of us, right?
He sure did.
Yes, sure did.
I adore this concept of parasites, pathogens, symbionts altering the behavior of their hosts. Sounds like you all do too. Elio had some interesting ideas about viruses and behavior. I know that folks are short on time, but the San Francisco writer, science fiction writer, coming back from San Francisco, right? David Brin has an old story about this called the Giving Plague, and he gives a link to it. Here you have a virus that causes altruistic behavior. It reminds me of the story, I'm sure it's not true, about the fellow who survives rabies. After he recovers, he is asked why he tried to bite people. He thinks for a moment and replies, "It seemed like a good idea at the time." Even though the virology may make you wince in the story, I presume. I love science fiction, but the authors sometimes, play a little fast and loose with the actual world of science. I think you and your readers might enjoy David Brin's story. I will be teaching a freshman writing course on symbioses and parasitism this fall at my small liberal arts college, and you can rest assured I will be discussing this.
Puget Sound University, by the way.
That's right. I will be discussing this and related issues with them, including TWiP. Happy for any suggestions or assistance out there. As always, I enjoy listening to your podcasts. Best wishes, Mark Martin. Thank you, Mark. This was actually received before he went there. Oh. The next one is from Tim, who is our farmer friend in Minnesota. He says, "Dear Racaniello, Schaechter, Schmidt, Swanson, thanks for the great episode on swabbing the environment around hospitals to check for prevalence of antibiotic-resistant microbes. The idea of resistance spreading out from healthcare facilities seems very intuitive after hearing about it, but I'd never thought of this before. It got me thinking about the number of other livestock farmers I know who have a family member that works in healthcare as well as helping on the farm. Could this be leading to increases in antibiotic-resistant organisms in livestock, or obviously vice versa?"
Ooh.
I bet.
Boy.
I bet, because,
I bet so too.
That study in the Netherlands where they went to supermarkets and looked at the chicken and what antibiotic resistance was there, and they found the same ones in the local hospitals. So I could imagine it going from farms to hospital as well.
Yeah,
sure.
or when farmers are hospitalized.
Sure.
They also would, yeah.
Or their farmer friends visit them.
Right.
Wow. The world's a scary place. I also found the practice of using antibiograms interesting. I had not known how doctors make decisions on which first-course antibiotics to use. I will have to ask my veterinarian friends if there is an antibiogram equivalent in animal medicine. I'm sure we have some vets listening who know. Thanks again for another great episode, and also for the TWiV bump for our Ag Side Today podcast. It was much appreciated by Steph and I. Hope that nice late spring, early summer weather has rolled in where you all are, and that you have a few chances to get out and enjoy it. It's a balmy 18°C here in Minnesota, and finally not raining for once, although it just did rain an inch and a quarter the other day, so we were due some decent weather. I'll also add a planting progress report. Despite that not being a typical feature on the show, we have about 60% of our hay fields seeded, 0% of our corn, and 0% of our pasture ground interseeded. Usually those numbers would all be around 100% at this time. It's been another wet spring, to say the least. You see, Elio, too much water in Minnesota and not enough in California.
I know, we could use it. And how?
Yeah, maybe you should go to... Yesterday when I was in San Francisco, someone said, yeah, we have no rain, and Los Angeles takes all the water that we have. Is that true?
Mark Twain said it well. Whiskey is for drinking, water is for fighting. They're very good at that in California.
Okay, that will do it for TWiM number 80. You can find it at iTunes and also at microbeworld.org/TWiM. And we love getting your questions and comments. Please send them to TWiM, TWIM, at twiv.tv. Michele Swanson is at the University of Michigan, which is in Ann Arbor, Michigan. Thank you, Michele.
Michigan.
Thank you.
Thanks for joining us today. And Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure.
And Michael Schmidt's not here today. He's in Washington, D.C. Hopefully you'll be here next time. I am Vincent Racaniello, and you can find me at my blog, virology.ws. You can also find me on Twitter, PROFVRR. I'd like to thank the American Society for Microbiology for their support of TWiM, and Chris Kindayan and Ray Ortega for all of their technical help. The music that you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.